Okay, let's resume from where we left. So we talked about polymerase chain reaction, how it works, the role of the primers, how you design them, and so on. And then we talked about one application in forensic studies. Now we're going to move on and talk about what other techniques have been developed out of spinning off from PCR. In particular, we'll focus on the quantitative real-time PCR. So throughout the 40 years that PCR, since the PCR has been invented, several applications have been built out of that technology. Here you just have a few examples listed here, but in reality there are many, many other ways in which PCR has been modified to suit different types of applications. For example, reverse transcriptase PCR or RT-PCR is a technique very commonly used to produce copy DNA or cDNA. It's basically a DNA copy of a mature mRNA. So an RNA without introns can be copied through this enzyme called reverse transcriptase from basically and converted from RNA, mRNA into a copy DNA. Why would you want to do that? For example, if you want to clone a gene, a coding sequence, obviously you don't want to clone all the introns and so on. So you can actually isolate the mature mRNA and reverse transcribe it into a copy DNA and then clone that cDNA, which then would be coding for an mRNA which does not include the introns. Other technique was this technique was also commonly used, for example, at the beginning to produce cDNAs for microarrays as well, to study gene expression in different tissue samples. Multiplex PCR is also another technique, another modification of PCR, which is used, in this case, fairly simple. It's basically like PCR, but you have multiple DNA primers used on the same sample, and this allows you to detect different possible or different targets within the same sample. For example, you can use it to detect different contaminants in a different type of, on the same type of tissue or food type, for example. And then another one is quantitative PCR, which is the one we're going to be focusing on from now on, or real-time PCR, qPCR, that's the way it's usually presented, which allows you to do not only qualitative but also quantitative analysis of DNA or RNA. So let's expand a bit on real-time quantitative PCR here. First of all, it's used to quantify, to determine fairly accurately the amount of DNA or RNA which could be present in a sample. DNA that could be coming from a contaminant microorganism, or RNA that could be coming from a contaminant microorganism. We'll see in one slide later on how this is the base for the current molecular COVID PCR testing. It has very high accuracy, very high sensitivity, so it really allows you to detect very small amount and determine their overall quantity with extremely high accuracy. So when usually it can be used both from DNA or from RNA. When you start from RNA, the RNA first has to be transcribed back into DNAs through the use of this enzyme called reverse transcriptase, and therefore in this case the quantitative PCR is referred to as reverse transcriptase qPCR, RT-qPCR. Some people omit the Q, but RT-PCR in reality is just a reverse transcriptase PCR. So it's not quantitative necessarily. So in this case, the quantitative PCR starting from RNA is reverse transcriptase quantitative PCR, so RT-qPCR. Now, how the base for quantifying the amount of DNA or RNA present in a sample is through the use of a fluorescent dye. Fluorescent dye who gets incorporated more and more into the amplicon, the more the amplicon is produced. So at the beginning will be very little or none, and then over time this amount will increase. These fluorescent dyes can be of different types. We will not spend time here really discussing, but could be either simply binding to the polymerized DNA, so to the amplicon, like cyber green is very, very commonly used, or could be also already conjugated to the primers, like TACMan here, which relies on FRET, but we're not going to talk specifically. But that's the fluorescence, increasing fluorescence that ultimately is followed through quantitative real-time PCR. So as the word says, real-time, okay, PCR means that you are actually following the production of the product in real time. How do you do that? Through, again, the integration of these fluorescent dyes and following the amount of fluorescence, how it increases over time through the different cycles. Okay. So now, as we discussed when we were talking about how PCR behaves overall, as we said before, there is a incubation time at the beginning where really there is no much DNA or RNA in this case, if we're talking about RT-qPCR, that is produced. Okay, and then certain point though, after a certain number of cycles, these amounts start growing exponentially. Okay, and that's through the exponential phase right here. Now, to a point where the DNA that previously was not detectable, so the fluorescence incorporated in the amplicon was so little that could not really be detected by the machine, becomes detectable. Okay, and then this goes on until you reach the plateau phase when you're running out of reagents. Now, this value here, the cycle through which you have that switch from non-detectable to detectable, is referred to as the quantification cycle, or crossing point or threshold cycle. Is the same thing. You may find different versions if you look online. I like the CQ word here. That's the most commonly accepted. This will be actually the standard name, quantification cycle there. So this is the cycle in which you reach the threshold. Okay, your sample has reached a value which now can be detected. Okay, and this is done automatically by machines. Okay, when you're doing the PCR. Now, this is the value that is key to quantify ultimately how much DNA or RNA was present in the original sample at the first cycle 0 here. How do you do that? Well, you do that ultimately by comparing the CQ value that you get, for example, in this case will be 20.6, whatever, to the CQ values that you will get with standard samples made by the same type of molecule. How do you do that? Well, at the same time, while you're running your sample, you will be running a series of samples produced by serial dilution of the same DNA or RNA molecule. So basically you have serial dilution. Let's say you start with a concentration 10 to the 5, whatever it is, the unit doesn't really matter. This will become 10 to the 4, then to the 3. So this gets progressively diluted, this sample. So you would do your real-time PCR on your own sample at the same time on standards. And this is what you will get. Imagine something like this. So these are the curves obtained with a standard sample, whereas the dotted one is the one obtained with your own test sample. These are all the CQ values, okay, of these different samples right here. You can see already that now the CQ value of your sample is in between these two values. So the concentration will be in between these two. Obviously, the earlier the CQ value, the higher the concentration. So these are the most concentrated sample, right? The concentration is going lower and lower. The lower the amount, the later on is the CQ value right there. So not only that, you know that it's in between these two values, but you can actually fairly precisely quantify by generating a standard curve with the standard samples, which correlates the CQ, in this case called CT, same thing, okay? So the cycle number in which the threshold is reached through to the logarithm of the concentration. In this case is DNA, but could be RNA as well. So these are the standard samples, and these are their CQ values, CT1, CT2, CT3, and so on, or CQ1, CQ2, call it whatever one. So this gives you a linear relationship. Okay, so if you just use the standard, you will get this linear relationship, which then would allow you to infer the actual concentration of your unknown sample here by taking its CQ value or CT value here, see where it crossed this curve, and this will give you the logarithm of the concentration of this sample. Okay, that's how, by following in real time, that's why it's called real-time quantitative PCR, By following the production in real time of the product of the amplicon here through fluorescence, you can actually quantify ultimately the amount of DNA or RNA. In the case of RNA, the sample would have first been transcribed through reverse transcriptase into DNA. Now, this is the common base, as I mentioned before, through which you do what is called the molecular PCR test, or molecular test for COVID, the most expensive one, just to give you an idea, the one that is somehow opposite to the antigen one. In the antigen one, all you detect is actually presence of proteins coming from the virus. In this case, with the molecular COVID test, what you do actually, you start from the RNA of the sample. So you first take a sample, as you know, in a very painful way sometimes, going to the nasopharyngeal area right here, or you can do it right at the nose. Now, use basically, if there is any virus, you put it in a buffer here, and then this RNA gets first transcribed back into transcribed to DNA. COVID is an RNA molecule, so it gets converted into double-stranded DNA, and then by quantitative real-time PCR with primer specific for 3 specific regions of the SARS-CoV-2 genome, you can actually detect whether in real time whether there is virus present ultimately in the nasopharyngeal cavity or not. What you will get in this case here is obviously the presence of a curve. If the virus is there, it will actually be 3 curves, because there are 3 regions that are investigated normally, and no curve if there is no virus there. Not only you can detect whether it's there or not, but based on the CT value that you get, and obviously these 3 curves should be fairly similar overall. Based on their CT value, you actually can determine how much virus is present, whether the load was very high or little in terms of COVID presence in the nose. Okay, let's finish up refreshing some concept of agarose gel electrophoresis here. So we already talked about electrophoresis in general in biochemistry, and as you know, it's a generic technique that can be used for DNA, proteins, and so on, that is used to separate molecules that contain a charge by putting them in an electric field. Now, it can be used for different purposes. Can be used to determine whether a molecule is there or not. For example, even by PCR, you can see whether a target is there or not, and therefore the genome that contains that target is present or not. So that will be just a qualitative type of analysis. But it can be used also to determine the size of specific molecules, and we'll talk about that in the last slide. Or even just to separate molecules. It's very common, for example, with DNA. After you cut, for example, a plasmid, you may have multiple fragments. Remember, we talked about restriction mapping, right? You can generate different fragments, and then you can actually cut out the desired fragment from a gel, purified, and therefore you would have only that specific fragment there. So it can be used for different purposes. Now, how does it work? No matter whether you're working with DNA or proteins, you are always taking these charged molecules and putting them in an electric field. Now, based on their charge, this molecule will be attracted by the opposite pole. However, in the absence of a gel, this would happen in a fraction of a second. And therefore, there is resistance that is created in the migration of this molecule by making what we call gels. These gels that can be made by different type of polymers, the common ones being agarose, which is is a natural one derived from seaweed, which creates size of pores that allow to separate only basically large DNA molecule, not smaller ones. Or you can use like polyacrylamide. It's an organic one. It's actually neurotoxic. It's got different properties there, but which allows for the separation of smaller DNA molecules or proteins as well. We talked about them in the context of protein, or when we're talking about the Sanger sequencing method of DNA, where you had very small molecule DNA with a 1 nucleotide difference. So you would have used a polyacrylamide gel. So what are the ways of separation? As with protein, the basal separation of any molecule in an electric field is based on three things. It's based on the size, overall size of the molecule. So the larger the molecule is, the slower it will be. The shape, okay, so two molecules with the same size but different shape would have different migration properties. The reason being that this shape would help some molecules to go through the pores better than molecules with the same size but different shape. And the overall charge to mass ratio. So the more charge you have, the more you're attracted by the opposite pole, even if you have the same shape and the same size. Now, when it comes to DNA, we talked about proteins, how SDS get rid of the shape and the charge to mass ratio. When it comes to DNA, in reality, the charge to mass ratio is not an issue. The reason being that the charge of each DNA molecules ultimately come from the phosphate group, right? And so since you have one phosphate group for each nucleotide, you can see that the more nucleotide you have, the more charge you would have. It would increase proportionally. So the ratio between the number of charge and the number of nucleotides will always be the same, right? 1, basically the ratio, right? So there's 2 per base pair, but for each single strand will be, so there will be 2 charge for each base pair, obviously. And so the more you have 4, you would have, so if you have 2 base pair, you would have 4 charge, 3 will be 6, and so on. So the ratio would always be 2 to 1 for base pairs, and 1 to 1 when it comes to single strand DNA or RNA, right? So the mass to charge ratio is not an issue for DNA. It doesn't influence the migration of DNA molecules. What influences the migration of DNA molecules is the shape and the size, right? And you can see how the shape affects the migration. We already talked about this, but just refreshing. When you're running a plasmid, so plasmid can exist inside the same bacteria in different shapes. Okay, so this is exactly the same plasmid, okay, and it has different level, what we call supercoiling. So it can be very relaxed, or can have more and more and more supercoiling. And now you can see here how if you run them into a gel, now the relaxed form will be slower than a supercoiled form. Now, how you can get rid of all of this is by basically giving the same shape to all the DNA molecules, and the way you can do it is just by cutting them. At that point, all of these molecules will become linear. They're all the same, identical, so they would have the same overall size. The charge to mass ratio is not an issue. They would all migrate the same way, and that's exactly what is showing you right here. So if you take this, you cut it, say with our restriction enzyme equal 1, let's say there is only 1, basically impossible, but let's assume it's 1, cut it here, 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 all of them will become linear, all of them would have the same shape, just same shape, same charge to mass ratio, and same size. They would all migrate at the same time. So digestion would help. In the case of PCR, obviously this is not necessary. When you are producing an amplicon, you're already producing a linear molecule of DNA. So in the example, for example, of the D1S80, you would have 2 alleles that are being produced by PCR. Both of them will be linear, right? They have the same charge to mass ratio. Shape is the same. The only way they will be separated in the picture that I showed you before is based on their size. Now, how do you prepare the Agarose gel? There's also a video under the lab that I post a series of videos. So the way you will prepare is first you would take the Agarose, and we go through this in the lab. You set a specific concentration based on the size of the DNA molecule you want to separate. The smaller the DNA molecules, the higher is the concentration of Agarose. So if you're talking about a few hundred base pairs, 200-300, you will go to 1.5, even 1.6%. If you're talking about plasmid DNA, easily you go with 1%. Agarose gel, even 0.8, depending how you want to separate them. And so you would then, to dissolve the agarose, you will see you need to actually provide heat, otherwise it just precipitates, it doesn't dissolve. Then you let it cool down, you add a DNA molecule to visualize the DNA, you pour it, and you cast it. Okay, you cast it and when you cast it, you create also those spaces called the wells, which is where you will be adding your sample. Once your DNA here has solidified with the wells present there, and inside here there is also a fluorescent dye that allows you to visualize where the DNA is, because the DNA would actually be invisible afterward. You can take your gel and you put it into an electrophoresis tank here with some buffer on top. You apply the current, okay, so negative current here, positive current. DNA would have a negative charge because of the phosphate group, so would tend to go to the positive side, so the wells will be on the opposite end. So you will load your DNA here, will be attracted and go through the gel. If you reverse this, then the DNA will be going out of the wells in a fraction of very few minutes. So how do you visualize DNA? You can visualize it in different way. You can use a radioisotope, so DNA could easily incorporate any radioactive form of phosphorus. So P32 is a very common way to make it visible. So you would just incorporate P32 in DNA. If you then put a film on top of that, wherever there is radioactivity, you will get a dark band produced there, and so you would visualize where the DNA was just by looking at the film. Or you can use fluorescence. Okay, so fluorescent can be either incorporated into the DNA or could be binding to the DNA, like with ethidium bromide, or in our case we will use red safe, which work the same way, which intercalate into the double strand. So these slides, that's exactly what it says. So these are fluorescent molecules. Ethidium bromide is the gold standard, if you want, for DNA staining in agarose gel. Now replaced by red safe, which, as the word says, should be safer than ethidium bromide, but I'm not 100% sure, because both of them, what they do, they intercalate between the two strands of DNA. So they actually go in between the two strands here, and so as ethidium bromide is mutagenic because of this activity, red safe does the same. I would still assume that red safe could be potentially mutagenic. So I would not take it as safe, as the name would say instead. So they do, both of them intercalate inside the DNA. And now they have the property that, as is, oh, they can look red and so on, but they're not really visible. And then both of them actually have the property that they can absorb UV light, and when they absorb UV light, they actually emit visible light. What does it mean, practically speaking? Is that if you take this gel here, right now you don't see any DNA, right? But there is either ethidium bromide or red safe present in there. And now this molecule here would have accumulated where there is DNA, because they intercalate inside the DNA. So while DNA is migrating, it will bind more and more and more ethidium bromide or red safe. So DNA is somewhere here, we don't see it yet. But now if you take the same gel and then you put it on a UV box, so there is UV light produced from underneath here. Now this molecule would bind the UV light, and when they bind it, they emit visible light. Since they are concentrated where DNA is, now where you see visible light at this point, that's where DNA is, and therefore you can visualize where the product of digestion or PCR are, or your standards are. Now let's finish here talking about how can you use agarose gel electrophoresis to quantify the size of a molecule, specifically DNA. Well, you do that by using what we call a standard or a marker. A standard or a marker is a mixture of DNA molecules that have a known size. Okay, for example, this marker right here is a mixture of DNA molecules of this size: 100 base pair, 200 base pair, 300 base pair. So you know exactly what they are. They're usually commercially produced. You can get actually a picture from the company that produced them that tells you exactly what the band should be. Okay, so you can actually map their size in the gel. Now, you can actually quantify any other molecule of DNA present in the same gel where you have put the marker by comparing its size to the one of the marker. You can get a gross estimate just by looking at it, like in this case, you know it's roughly 300 base pair, but you can have a much more accurate estimate, particularly when these molecules are in between these. Their migration is not linear, as you can see here. It's always 100 base pair difference, but you can see that the distance here of migration is different, right? So you can do that, particularly when it's in between, by generating a standard curve using your marker. So you know the size of all these molecules. All you have to do is actually calculate or measure how far each of these molecules has migrated from the place where you loaded them, so the well. So this, for example, could be, let's say, 6.2 centimeters. This will be 5.7 and whatever you measure, and then you can then correlate these two, so the size, or better, the logarithm of the size and the distance of migration, to generate a standard curve. Here you can see on the x-axis we have the distance of migration. Imagine this molecule being these ones right here, and this is the logarithm of the size of the fragments. As you can see, the larger the molecule, the smaller is the migration. You can see right here, 800 base pair migrates less than 100 base pairs, assuming that the wells are here. So once you generate this standard curve, now with the standard right here, now you have a relationship established between the size, the logarithm of the size in base pairs, and the distance of migration of the bands, right? So anything else that is in the gel, and it has to be in the same gel, right? Because this distance would change depending on the time you let it go. So but any other molecule that is in the same gel now can be quantified by just measuring how far it has migrated from the well, right? And then once you know the distance, say this is for something, 4.2, you just see where it actually crosses the curve, and then you can figure out the logarithm of the size of that molecule. If you do the antilog, you will actually have a fairly accurate estimate of the size of this band. So summarizing everything that we have discussed in these two parts of this lecture. First of all, we went through PCR. The main focus was talking about PCR and the modification of PCR. So we talked about how PCR works. We talked about how mechanism of action, the role of DNA polymerase, and all the other components that are present in the PCR reaction. And then we focused specifically on the role and how particularly primers are designed. How you have to consider the length, has to be between 18 and 24 nucleotides, if you remember. You have to avoid the formation of hairpin or cross annealing between the primers. The primers have to be matched, if you remember. So they should have a GC content between 60 and 40%, and therefore similar melting temperature we do to the two primers, melting temperature that you can estimate grossly based on the GC content. And then once you have the melting temperature, you can figure out the potential annealing temperature of these primers by subtracting 5 degrees to the from the melting temperature. Then we went through some example, or we talked about one example of how PCR as is can be used in forensic studies, and we talked about the D1S80 locus, which is a variable number of tandem repeat type of locus. There are several of them again in your genome, it's just one of them. And we discussed how this can be used ultimately to determine the identity of an unknown DNA coming, for example, from a crime scene. And then we talked about modification of PCR. We talked about how there are different ways in which PCR has been modified. We talked about reverse transcription PCR very briefly, multiplex PCR, and then we focus on quantitative real-time PCR. We talked about how the quantitative real-time PCR works. First of all, by using fluorescent, you follow in real time the production of a specific molecule, of an amplicon starting from a specific target, right? And by following that, as soon as this molecule becomes detectable based on the fluorescent, now that value, that cycle where this molecule becomes detectable is the cycle that holds the information to quantify that molecule by comparing to standard sample. Then we finish up talking about refreshing some concepts about agarose gel electrophoresis, how it works based on charge to mass ratio, size, and shape, where we discuss also how charge to mass ratio is not an issue in DNA. And then we talked about how the shape can be brought to be the same by digestion when we talk about large molecule, but it's not an issue when we talk about PCR amplicons, for example, because they're all linear molecules. And then we finish up talking about how we can use the information on an agarose gel to actually quantify the size of any molecule present in